Experts say genomic sequencing is crucial in disease response. Why is this aspect of science so important, and what does it mean to the future of public health? Today on Soundhealth, we will find out. I am Olasumbo Modupe. Thanks for always staying with Lagos Television. The program continues in a moment. Please stay with us.
Genomic sequencing is a process for analyzing a sample of DNA taken from your blood. In the lab, technicians extract DNA and prepare it for sequencing. Within every normal cell are 23 pairs of chromosomes. Chromosomes are structures that house DNA. DNA is coiled into a shape called the double helix. The double helix can be unwound into a ladder shape. The ladder is made up of paired chemical letters called bases. Altogether, our DNA contains about 6 billion such paired chemical bases. There are 4 bases in the DNA alphabet: A, T, C, and G. In DNA, base A joins only with base T, and base G joins only with base C. To read the sequence of bases in DNA, samples are inserted into a sequencing instrument, where high-frequency sound waves break the DNA into smaller pieces that are only about 600 bases long. Special tags are added to the ends of the fragmented DNA. These tagged strands of DNA can then attach to a glass slide. In a sequencer, each piece of DNA is copied hundreds of thousands of times, which creates clusters of identical DNA fragments. Next, the sequencer reads the DNA, one base at a time, using different colored tags for each DNA base. Special sensors within the machine detect the different colored tags. This sequence of colors reveals the DNA sequence of each fragment. Powerful computers piece together these individual DNA fragments and reveal the sequence of your DNA. Then a team of medical experts uses specialized software to analyze and compare the DNA sequences to identify the handful of variants that may be important for your medical care.
It is still Sound Health on Lagos Television. Joining us via Zoom this evening is Dr. Oladipo Kolawole, Head Department Microbiology Adeleke University, and founder Helix Biogen Institute, which has developed COVID-19 vaccine candidate that has been captured by the World Health Organization, making it the second in Africa. Good evening, Dr. Kolawole, and thanks for always obliging us.
Oh, good evening. It's my pleasure to be on this platform for this period of the time.
Okay, Dr. Kolawole, what is the exact role of genomic sequencing, and why does it really matter to the future of health, public health?
Now, from the root word, gene, that's why we have genomic. And when we talk about gene, that means that we are talking about the genetic makeup of an individual, either animal or organism. So when we now say genomics, it's just the study of the composition, the function, the differences, the similarities in a gene of an individual or of an organism, whereby to get more detailed information about those genes. And like we have really experienced in some couples of months ago, we can see Omicron, Delta, like that in terms of SARS-CoV-2. Those are the genomic sequences that enable us to classify those pathogens according to their lineages or sublineages. And even there was some couples of months ago that we hear that a whole genome sequencing of human was done. That enabled us to have a full understanding about the genetic makeup of a human. So it is very, very crucial and uprising tool in health sector now in order to understand how we can prevent a disease and how we can cure or treat a disease condition in an individual. So genomics is an interesting tool that is highly appreciated in healthcare delivery these days.
All right. Looking at trends in countries, especially Nigeria, do you think this tool is being utilized well enough?
Yeah, we have the usage of the tool in different ways. We can use it in responding to epidemics or pandemic, and which we had seen since 2019 that Nigeria has actually used this tool in other. NCDC is using it. NIMA, Nigeria Institute of Medical Research, in Yaba, Lagos, there they have been using it. HSheet have been using it. 54 Gene have been using it. So the other aspects of usage of genomics is what we call translational research in development of products. And I think that it's only 54 Gene in Lagos that is working in this pipeline in this way in order to develop some certain drugs. And they are concentrating on communicable disease, targeting cancer, various kind of cancer, either prostate or breast cancer, things like that. So Nigeria has not been using so much in that line, but in terms of pandemic or epidemic responses, we have actually used the tool to solve those challenges we have in terms of health.
From the picture you painted now, countries are using this tool to detect diseases. I know we've reported more cases of Ebola, COVID-19, now hepatitis, as well as monkeypox. Do you think we are towing this part, or we are just living on assumption just because some cases are reported in some other parts of the world?
Yeah, we are showing the part because we also are reporting based on what we are actually seeing. The only challenge we have is that the capacity is very low compared to some other countries because, like I said, some couple of days ago, that even some of the machines cost up to 600 million naira.
Yeah, genomic sequencing is not well utilized in Nigeria like other parts of the world, maybe because of the cost implication. Like I've earlier said, that a machine costs something like 600 million naira in terms of Illumina for NGS. So we need to use this technology more, though the cost implication is there, and importation of reagents to do that because it's not locally sourced. So, but it has been of great importance and help. COVID-19 have shown the significance of this genomic sequencing.
Well, in your response, you mentioned that funding is a major challenge to genomic sequencing in Nigeria. Do we have enough expertise? Do we have enough professional to man these aspects of science?
Yes, we have expertise, but not as much like compared to other countries. And I know that a series of capacity building has been done in such regard in order to train as many scientists to know how to use the equipment, even to the latest one that we call a nanopore sequencer, which is just a mini one that can be used, not as big as Illumina and the other generation of sequencer. So we have expertise in the country that can handle that.
Dr. Kolawole, many have argued that rather than focusing on genomic sequencing to check diseases emergence and prevention, shift should be on public health care settings. Do you share the same thought?
Looking at primary health care, yes, it may be difficult to adopt it in primary health care because most of our primary health care do not have light facility, storage of some reagents to use for it. But still, we need to complement each other. As we are using the former method of diagnosis, using to cater for diseases, we can use the new technology too, so that we use the new and the old school together. So each other complement each other. Some have a disadvantage where the other has disadvantage. So we can make up for each other in terms of these aspects.
How do we strengthen these aspects of public health?
Another way that which we can structure and strengthen the structure of these in public health is that we need the government to buy more of this sequencer. I know NIMA, I've talked about NIMA, I've talked about HCheat in Redeemer, I've talked about NCDC in Abuja, that they have the capacity. Those are the few 154 those are the few ones that I know. Maybe there are some other labs in the country that I didn't know, but those are the few ones that I know. But they are not enough, they are not sufficient. So we need to put in more money into these genomic sequencing so that we can have the full information of any organism in question we actually are interested in.
Okay, Dr. Kolawole, as we wrap up now, what is your call to action?
One of the calls to action is that we need private individuals to invest into our health sector. The government cannot do it alone. So, and all the three tiers of government, at the local government, at the state level, at the federal level, should have some role to play in this health, so that we can have enough information about any organism, and this can easily help us to prevent future occurrence of any outbreak of diseases in our community. So invariably, this is a tool that other countries are using, and we too can adapt those tools to our health sector, and we're going to gain more from it.
Dr. Oladipo Kolawole, Head Department Microbiology Adeleke University and founder Helix Biogen Institute. Thank you so much for your time and expertise. We really appreciate your thoughts on the role of genomic sequencing to the future of public health.
Thank you for this privilege.
All right, I have been speaking with Dr. Oladipo Kolawole, Head Department Microbiology Adeleke University and founder Helix Biogen Institute on the role of genomic sequencing to the future of public health. Sound Health continues with Trending Health Report. Please stay tuned. It is still Sound Health on Lagos Television. More to come in a moment. This is where we say thank you on today's episode of Sound Health. Remember, diseases are real and we can only check the spread of diseases by protecting ourselves by adhering strictly to all precautionary measures. Protect yourself and your loved ones. Wash your hands for at least 20 seconds with soap and water or alcohol-based hand sanitizer. You also need to get vaccinated. The vaccines are safe, free, and harmless. For comments and inquiries, please send SMS to 0035826603 or follow us on social media at LTV Social #SoundHealth. A sound health is a sound mind. Make healthy living your choice.